biological waste and the different types of things which we are doing while carrying on the routine procedures in our labs. So there is a specific decontamination requirements which will depend on the type of the experimental work and the nature of the infectious agents which are being handled. So according to that, we have different levels and different types of sterilizations and disinfectants which we are using in our labs. And there is a particular contact time for which you need to have the disinfectant for specific material and for specific according to the manufacturer which is there. So, moving on to basic definition, sterilization is defined as a process by which an article, a surface, or a medium is freed from all living microorganisms, either in its vegetative state or spore state. So, here we need to understand the difference between sterilization and disinfection. By disinfection, we are meaning a process which is physical or chemical, which means to destroy the organisms but not necessarily the spores. So here, as you are noting, it has been underlined and shown to you that the difference between sterilization and disinfection is that in sterilization you will also be destroying the spore state of the organism, whereas in disinfection you will only be dealing with killing of the microorganisms. Then the third term we have is asepsis, which is the term used to indicate the prevention of infection by preventing the growth of bacteria in a particular wound or tissue. This is the process which you usually carried out before drawing out a blood sample or before and performing any surgical procedure. Then the fourth term we have here is antiseptics. It is defined as a substance which inhibits the growth and development of microorganisms without necessarily killing them. So antiseptics are usually which you are applying to the body surfaces. Like once you get hurt or something, you are applying Dettol or Savlon for the regular cleansing of your wounds. That is in that state, what you are using is called an antiseptic. Then we have decontamination. Decontamination is the process of removing or killing of microorganisms. The same term is also used for removing or neutralizing your hazardous chemicals and radioactive materials. And then we have cleaning. Cleaning is the basically physical removal of dirt, organic matter, and stains. Cleaning includes your brushing, vacuuming, dry dusting, washing, or damp mopping with water containing a soap or detergent. It is the regular procedure which is carried out in your homes and everywhere in your environmental settings. And pre-cleaning is essential to achieve proper disinfection or sterilization, because once you are not mechanically removing the waste, then you are not able to properly decontaminate or disinfect the particular substance. So first you go about cleaning and then you go about disinfecting or sterilization of the particular thing which you are using. Next, we have bactericidal and bacteriostatic agents, which will be discussed further in details in the next coming slides. We have different methods of sterilization and disinfection, which includes your physical, chemical, and physiochemical methods. These are the broad differentiation of your methods. In physical methods, we are using a physical form of energy to disinfect your or sterilize your particular entity. It includes your sunlight, heat, vibration, radiation, and filtration. In heat, it can be of two types again, dry heat or moist heat. In dry heat, we have red heat, flaming, incineration, hot air oven, and infrared radiations. In moist heat, it has been differentiated according to the basis of temperatures. You can have a temperature of below 100 degrees Celsius, at 100 degrees Celsius, or above 100 degrees Celsius. In radiations, they can be of broadly two types, non-ionizing and ionizing radiations. In your ionizing radiations, you have electromagnetic and particulate radiations. In the filtration, you can use earthenware, asbestos-based filters, sintered glass, or membrane pore filters. Then the second category is your chemical disinfection methods. These include your liquid and gaseous state. In liquids, which we are using in our day-to-day practice, like sanitizer, which are alcohol-based rubs, you can use aldehydes, phenolic compounds, halogens, heavy metals, surface active agents, and dyes. In the gaseous forms, it is formaldehyde, ethylene oxide, and plasma. And in your physiochemical methods, we'll be discussing them. They are relatively newer methods which are used for sterilization. So we'll be discussing them separately. Then you have the physical methods. Then first method which you are using is your sunlight. Sunlight is basically used for your spontaneous sterilization of materials. It has a bactericidal activity. There can be two types of activities for any sterilization or disinfection agent. It can be bactericidal. It means it will be resulting in the elimination or killing of that bacteria completely. And it can be bacteriostatic. Static means the bacterial growth will be stopped at a particular level. As you all have studied about the bacterial reproduction curve, it has 4 phases. So it will put a stop to that phase and it will not let the bacteria reach the log phase of multiplication. That is your bacteriostatic activity. Apart from this, we have natural UV rays and heat rays which are used for your sterilization or disinfection purposes. And we have drying, in which is infective to spores. So hence it is acting as your disinfectant method. Then heat is the most common and reliable method which we are using in our day-to-day practices in our routine lab also. So dry heat basically works on the principle of denaturation of your protein structure of the microorganism. It leads to an oxidative damage and toxic effect of the electrolytes. Moist heat leads to your denaturation and coagulation of proteins. Matlab, in addition to your denaturation, in the moist heat, it is also causing coagulation of your proteins. And if we are using steam as a method of disinfection, it is using your latent heat of sterilization or evaporation for your disinfection. You all will be very conveniently knowing about, I don't know, 1 month or so we had the practical classes, and everybody of you who were preparing a gram stain for your practical, you were actually carrying out the dry heat methods of disinfection, because once you were picking up a slide, you were passing it over the flame and you were actually decontaminating the slide and removing all the visible dirt and grease so that you could make a smear on the slide. So you were actually carrying on the process of disinfection of that slide. And then you were using a loop which was heated till it was red hot to place a drop of bacterial colony or the normal saline which we use as a drop for preparation of gram stain. So that was also actually a process of disinfection. So dry heat basically includes your flaming, in which we are using inoculation loop and wire or your tip of the forceps. And the second method is incineration, and third is hot air up. So incineration is a method of sterilization where a very high temperature of approximately 800 to 1000 degrees Celsius is achieved. It leads to burning of volume. So obviously, when you are heating a temperature to such a high, heating a substance to such a high degree of temperature, you are reducing the weight and volume of your waste. The volume of waste is actually reduced up to approximately 10 to the power 4 to 10 to the power 5 times, so that your actual bulk of the waste which is generated is rendered less and it can be easily disposed of. So we have various types of incinerator. They can be cottage incinerator, single chamber, double chamber, or this is basically according to the design of the incinerator which we are using. Important thing which needs to be remembered here is by common sense that obviously plastic cannot be incinerated because it leads to generation of toxic fumes. Then this is basically a picture of incineration machine which is shown to you. It has various parts in it. You can see in the picture it has a cover, then you have a sampling port, then you have a thermocouple, then you have a big area in the center which is your which has a combustion and a control panel in it. There are air ducts present in it to regulate the flow of air, and in the middle where the area is getting red hot, this is actually your incineration chamber where your higher temperatures of 800 to 1000 degrees Celsius are obtained. And then as you can see in the outlet area, the incineration door is there. And here, from here, you can take out the material which has actually been reduced in the volume, and you can obtain it in the form of ash, basically, incineration ash. Next, we have a hot air oven, which is an important short type of question for you guys. It is commonly asked in your theory exams as well as in your practical exams. In the practical exams, when you are provided with a grand viva tray, there are various things kept like your tuberculin syringes or your petri dishes or your agar plates. anything which can be which you pick up can be asked about, and then the next obvious question is how are you going to disinfect or sterilize this particular equipment or this particular thing in your laboratory setting. So this is important for you guys. So hot air oven is the most common method of sterilization by dry heat. Holding period is the thing which you need to remember for this. It is 160 degrees Celsius for 2 hours. It is basically used for disinfection of your glasswares, forceps, scissors, and liquid paraffin. These are the most common materials which we are using in our routine microbiology laboratories. So it is loaded and cooled slowly so that the temperature is reached gradually, and once the temperature gradient is maintained, it is cooled down slowly so that the things which we are heating, basically the glassware, they do not crack. Because once these things crack, they cannot be used for further purposes. Then for hot air oven, we have a sterilization control. So sterilization control again can be divided into three different types. Either it can be biological, it means some living microorganism which will act as a control for the sterilization process, or it can be a chemical indicator like based on the pH change or the gas which is produced, which is changing some sort of chemical production, which is eventually visible to us in the form of color change, or it can be a physical method. So in biological methods, we are using a spore of Clostridium tetanus, which is a non-toxigenic strain of it. It is placed on a strip, and this spore strip is placed with the load. After processing this, it is kept. The process which is over, we kept it and incubated in the Robertson cooked medium at 37 degrees Celsius for 5 to 7 days. If in the anaerobic medium, the spores of Clostridium tetani or the bacteria is growing, then it is an indicator that we have not achieved a proper sterilization and we need to reevaluate the process. The second indicator is a chemical indicator. It is a brown tube which turns into green color if it's a positive test. And the third physical indicator which we are using is a thermocouple, which is a temperature sensitive instrument which changes the according to the temperature which we are. So this is a picture of the hot air oven. Most of you maybe have seen it in the practical hall or somewhere in our laboratory. So here we have different shells in which we can place our equipments for sterilization, and above it we have a panel on which we have a temperature control and a time control which we can set according to our requirement. And on the sides we'll be having a small fan which is there for circulation of the air all over the materials which we have placed inside this. Next method of sterilization is your moist heat, which is based on the temperatures of less than 100 degrees Celsius. So we are done with the dry heat methods of sterilization. Now we are moving to the moist heats method of sterilization, which we had as earlier studied in the picture which I have shown you, which is divided into the three different types of temperatures based on the range, which is less than 100, at 100, and more than 100. So starting with the less than 100 temperature, as you will be already aware of this well-versed technique, which is pasteurization. This is important for your the milk factories. This is used, and two methods of sterilization is there. One is Boulder method, in which you have to hold your particular samples for disinfection at 63 degrees Celsius for 30 minutes, and other is your flash process, in which you have to hold it for 15 to 20 seconds for 72 degrees Celsius, followed by cooling to 13 degrees Celsius. So this is destroying all your known spore forming pathogens such as MTB, Brucella, and Salmonella. These three are basically your foodborne or milk-borne pathogens. And pasteurization is a very common process which we are using in your milk industry for sterilization and packaging of your milk-based products. We can also use this for vaccine bath. Vaccines are kept at a temperature of 60 degrees Celsius for 1 hour for sterilization. And it can also sterilize serums at 56 degrees Celsius for several consecutive days. Serum are used for various tests in your microbiology laboratories. The second method here is inspisation. Inspisation is a method which is commonly used for your various non-agar based medias like LJ medium or the serum slope medium, which is a loftless medium. As you will recall from your earlier classes, LJ medium is the culture medium which we are using for culture of MTB, and your loftless serum slope is the medium which we are using for your culture of Corynebacterium diphtheria. Both of these are the mediums which are sterilized using inspisation. They are not agar-based mediums. They are solidified. One is an egg-based medium and one is a serum-based medium. So we are not using your regular methods of sterilization, which we are using in the case of agar-based mediums. So here we heat at 80 to 85 degrees Celsius for 30 minutes for 3 consecutive days. So this is a protocol of heating on 3 consecutive days so that you can destroy the spores which are formed. Like once on the first day you are heating, some spores will germinate after that, then these will be destroyed on the next day. Then on the second day when you are heating again, after this process also some remaining spores will germinate, and again on the third day. So basically you are giving the time gap for the bacteria to germinate the spores, and then you are subsequently heating it for 3 days and destroying all the spores which are formed. The second, we have temperature at 100 degrees Celsius. The most common example which we are using in our household settings also is the boiling. It is not recommended for routine sterilization of instrument. And for this, we have to make sure that we are not using hard water. So for that, we can add 2% sodium bicarbonate in the water to so that the water loses its hardness and it is converted to soft water. We can also use steam at an atmospheric pressure of 100 degrees Celsius. For that, we have a instrument which is called as Cox or Arnold steam. Then we have a process called tyndallization and intermittent sterilization, in which we are using your media containing sugar or gelatin exposed for 100 degrees Celsius for 20 minutes for 3 days. Again, the same principle that we are heating for consecutive days to destroy the spores which are formed. The third is when you are using steam under atmospheric pressure of 100 degrees Celsius. For that, we have various instruments available like laboratory autoclave, hospital dressing sterilization, bowl and instrument sterilizer, and rapid cooling sterilizer. So autoclave, this probably you would have seen in your various labs, or this big autoclaves are actually present in the hospital settings also. They're the most commonly used sterilization instruments which are used in your microbiology or your day-to-day practices. So what is the working principle of autoclave? Whenever you want to understand the working principle for an autoclave, you just have to imagine a pressure cooker. It is working on the same principle as your pressure cooker which works. So water boils when its vapor pressure is equivalent to that of your surrounding pressure, and hence when you are closing the lid and a particular amount of pressure is attained inside a closed vessel, the temperature at which the pot boils will increase. This is according to your law, which states that the pressure and volume is directly proportional to your temperature. So once you are raising the pressure inside a closed volume, the temperature in it will also increase, and the saturated stream which is generated, it has a penetration power, and it will penetrate the instruments which you have kept inside your autoclave, and it gives a latent heat of vaporization. Here are some stats for that: 1600 ml steam at 100 degree condenses in a 1 ml of water, and it releases about 400 calories. So whole of this calorie is basically converted into your steam, and the steam is basically vaporizing your acting on your instruments which you have kept in the autoclave and used for sterilization of these instruments. So as you can see from the structure here, you can see there is a pressure regulating device, as you have that on your pressure cooker also. You have a gauge which will indicate your pressure in the atmospheres, in which is the amount of pressure which has been received. You have a safety valve over here which goes off when the pressure exceeds the limit which you have set. Then you have a lid which is used to close this whole instrument. Then you have a handle for operating the lid. Then you have the body of the autoclave. There is a steam release valve and a vacuum release valve, and it is all of this instrument is balanced on a stand. So similarly, you also have various types of autoclaves according to the design on which they have been built. They can be gravity displacement, they can be horizontal, they can be vertical, they can be high vacuum sterilizer, they can be steam flash pressure pulsing autoclaves. So this is a diagram basically showing you the working principle of your autoclave. This is basically a horizontally designed autoclave, as you can see by its position. So you can see there is a door, there is a steam chamber. The equipment which is placed here is a conical flask. There is an exhaust valve present on the top to remove your steam after sterilization, and there is a waistline from which the water which is condensed has come out. So basically, you are heating. You see a pressure regulator for steam supply. The steam supply is coming from the below. We have a thermometer to read the temperature, and we have a sediment screen and through which air is passing. So once your steam which is generated passes through the passage which is shown here, marked in red arrows. It is perforating your shelf. It is coming onto your equipment. It is working on this, and then there is an operating valve which is controlling your steam, which is coming from your jacket to the chamber, and we have an exhaust valve from where, after the steam has acted on your particular equipment, it is leaving from there. Again, we need a sterilization control to check whether our equipment is functioning optimally or not. As you would have remembered, in the hot air oven, we were using spores of Clostridium tetanus. Here we are using spores of Bacillus stereothermophilus, which is 10 to the power 6 spores are used, which are placed in a paper envelope, and strips are inoculated into the suitable medium and again incubated at 55 degrees Celsius for 5 days to check whether these spores are germinating or not and whether our sterilization has been achieved optimally or not. Again, a chemical indicator, which is the same indicator, which is the brown tube, which changes to green color in the presence of the for control is used. Then the same physical methods we are using here is the thermocoupler. Then we have a process called as flash sterilization, which is an emergency. As your name suggests, flash word suggests that we are using in case of emergency settings. So this is an emergency procedure in unwrapped devices which are exposed to pressurized steam for 3 minutes. It is basically used in your OT settings where in an emergency procedure you require a particular set of equipment. The sterilization control here again is Bacillus thermophilus, stereothermophilus in a steam, and it ensures your sterility level is achieved. Then moving on to your filtration. So we are done with your physical methods in which we studied about heat, and we studied about the different temperatures at which we can carry on the process of disinfection and sterilization at moist heat, and the other we studied was dry heat. So now moving on to filtration. Filtration removes your microorganisms, and your heat labile liquids can be used for sterilization by this process, like your sugar solution and serum. It basically uses 3 types of filters. We can have a candle filter which has a all grid porosity, and you can use a ceramic filter or you can use a diatomaceous earth burr field filter. And you can also use a asbestos filter. We can use a which is for example of which is Seeds filter. Absorbing capacity is good, and it can alkaline the filtered liquid. Seeds filter is also available with us in the lab, and whenever you'll be joining back with your practicals, we'll show you this. This is a part of your grand viva tray, and you have to recognize this instrument, which is your seeds filter. Then we have a membranous filter, which is a cellulose ester filter. It has a specific pore diameter of approximately 0.25 millimeter. This is your filter, which is basically used in your filtration of water settings, like in your AquaGuard or RO based settings. This is the membrane type of filter which we are using for filtration of water. So you all would have seen a basic filtration apparatus in your chemistry times in your 11-12 standards. Here we have a filter paper cone, and we have a funnel, and we have a ring for support. The solvent which is containing your insoluble material is poured into it through your filter paper cone. The filter paper filters it out, and we obtain a solution with no impurities in your flask. Then moving on to the next method of sterilization, we have radiation. It can be non-ionizing like infrared and UV rays, or you can have ionizing radiations which are gamma and high energy electrons. There is, in case of ionizing radiation, there is no appreciable increase in temperature, so it is referred to as cold sterilization method. For example, we are using it for sterilization of plastic, syringe, swab, catheter, fabric, metal foils. We are using all this because for this plastic purposes, we cannot use your heating methods for sterilization. So they are basically sterilized by your ionization methods. As you will see that all the swab syringes we are used, they are basically disposable and one-time use. But if you would see on that wrapper which is there on that syringes of yours, it is always written that it is sterilized by your gamma radiation. Then moving on to characteristics of an ideal antiseptic or disinfectant, they should have a wide spectrum of activity, and they should be effective against all microorganisms, which include your bacteria, including your spores, viruses, protozoa, and fungi. They should be active in presence of organic matter. They should be effective in acid as well as alkaline medium. They have a speedy action. They have a good penetrating power, and they should be stable. So these are your ideal antiseptic or disinfecting agents. So these are the basic properties which should be present in every basically antiseptic or disinfectant to ensure that its functioning is to an optimum capacity. Then they should also be compatible with other antiseptics, and they should not corrode metals. They should not cause any local irritation or sensitization of the skin. They should not be deodorizing. They should not interfere with your healing. They should not be toxic if absorbed into the circulation. They should be readily available and inexpensive. They should be safe and easy to use. So the factors which determine your potency of disinfectants include concentration and stability of the substance. Like we have your sanitizers which are available in your various alcohol concentrations varying from 70 to 60 to 80%. The time duration of exposure which is required for that particular disinfectant to act, the particular pH, temperature, nature and number of organisms which it is going to act against, and the presence of extraneous material which is there. So mode of action, the chemical agents act in various ways. They act by destruction of cell membrane, resulting in exposure, damage or loss of contents. They can be surface active disinfectants, they can be your phenol-based compounds, or they can be alcohols. Or they can act by protein coagulation, which was how your heating methods were also working. This also includes some alcohols, acid and alkalis. They can also act by removal of free sulfahydryl groups, which are essential for functioning of some enzymes. And they can also act by competitively interfering with your substrate, enzyme substrate complex. So chemical agents which are commonly used as antiseptic disinfectants include alcohol, the most commonly used disinfectant in nowadays settings. It includes your ethanol, isopropanol, benzyl alcohol. Ethanol is your most common sanitizer-based products which we are using. So it acts by denaturing your proteins and make lipids soluble. Its level of activity is intermediate and it is used basically as a skin antiseptic. So here we are determining the level of activity as low, intermediate and high according to the spectrum of diseases and spectrum of microorganisms for which it is effective against. Like if it is effective against virus, bacteria, fungus, or various other organic compounds. Next, we move on to halogens, for which we have tincture of iodine, which is 2% iodine and 70% alcohol, and a povidone iodine. Povidone iodine, you would be aware of, is used as a common disinfectant before surgical procedures. It is also acting in the same mechanism by inactivating proteins. It has tincture of iodine has a low level of activity, whereas povidone iodine has an intermediate level of activity. They're again, as I already told you, used as skin antiseptics. Then we have detergents, which are basically your quaternary ammonium compounds. They act by disrupting your cell membrane. Basically, they act as your cell membrane destabilizers. They have an ionic moiety which interacts with your various components of your cell membrane and leads to disruption of your cell membrane synthesis. They have a low level of activity and they are also used as skin antiseptic and disinfectants. Then we move into phenol-based compounds, which we have phenol, carbolic acid, lysol, and hexachlorophene. Lysol, as you would have already heard of, is commonly used as a disinfectant for cleaning your floors and wiping off your various laboratory slabs and your working areas. They are also working on your same principle of denaturation of proteins and disruption of your cell membrane. They have an again intermediate to low level of activity. We are using it for disinfection of floor, wall, table surfaces which are at higher concentrations and used in soaps in our daily cleaning practices at low concentrations. Then we can also use heavy metals, silver nitrate, which is AgNO3 1%. As you would have, I guess, know, silver is used basically as your dressing in your wounds and burns areas. And we can also use mercuric chloride, which is HgCl2. They again precipitate protein and they react with SH groups and lead to lysis of cell membranes. They again have a low level of activity. They can be used as an eye drop solution 1% and disinfectant and toxic at higher concentrations. Then we move on to aldehydes, which is formaldehyde 8% and butyldehyde 2%. It reacts with your NH2, SH and COH groups. It has a high level of activity. It is sporicidal and toxic to humans. Then we have your gases, in which we have the most commonly used gas, which is ethylene oxide. It is an alkylating agent and it is a very high level of spectrum of activity. It is again used for sterilization of heat sensitive instruments. As I've already told you that the principle of heating instruments is limited to particular set of instruments. You cannot use certain instruments which are liable to get damaged by your heat product, so that we can use ethylene oxide as a method of sterilization. and it has an additional advantage of killing the spores as well. So, recommended dilutions of chlorine releasing compounds. We have a different set of conditions, whether the environment in which it is working is clean or dirty. Obviously, a higher concentration of compound will be acting in your dirty conditions, and a relatively lower set of lower concentration of compound is required in case of your clean conditions. So available chlorine required 0.1% or in clean conditions, and 0.5%, which is 5 gram per liter, is required in your dirty conditions. According to that, we have various formulations like sodium hypochlorite, which is available as 5% chlorine, calcium hypochlorite as 70% chlorine, sodium dichlorosuccinate as 60% chlorine, and sodium dichlorosuccinate tablets in your 1.5 gram tablets formulations, and chloramine in your 25%. So basically, this is your percentages showing your active chlorine gas which is there in your particular disinfectant. Then the various disinfectants which we had already studied, here we are summarizing it again. They are alcohol, aldehydes, phenols, in which some act as disinfectants like cresol, lysol, orthophenol, and some act as antiseptics like your Dettol and your Savlon, which is containing your chlorhexidine and chlorzinol. Then we have halogens. We have iodine in the form of tincture iodine and iodophore. We have chlorine in the form of gas and various tablet and hypochlorite formulations. We have heavy metal derivatives, basically your silver derivatives. We have your surface active agents, which are basically your detergents, which are basically acting as your membrane destabilizers by formation of cations, anions, and amphoteric compounds. Then we have your dyes, which can be aniline or acridine dyes. Then we have your gases, which are ethylene oxide, formaldehyde, and beta propyl. Then alcohols can be bactericidal, fungicidal, but they are never sporicidal. So, as I already told you, 70% ethyl alcohol is your concentration which we are using in your sanitizers, and isopropyl alcohol has a lower concentration of 35%. They act by denaturation of your bacterial protein. Then we have your formaldehyde, 40%. It is employed in liquid and vapor states. Formalin, which you will be well aware of as you would have attended your anatomy classes, and the preservation of specimen also in case of your PATH classes. So we are using a 37% solution of formaldehyde. We can also use a paraformaldehyde, which is a polymer containing 91 to 99% formaldehyde. So it is used in aqueous solution as bactericidal and sporicidal, and it has lethal effects on viruses, but lesser effect on spores. 0.2 to 4% formalin is used to inactivate viruses, which we can also use for preparation of your vaccines. Basically, the preparation of your killed vaccines in which we are killing your particular virus, it is can be done with the help of formalin. And we can also use a solution of 10% formalin and 0.5% sodium tetraborate to sterilize your clean metal instruments. Then next, moving on to glutaraldehyde. Again, when you will be attending your practical classes, we also have this Sidex bottle, which is available as a thing in your Grand Viva tray, and we show it to you in your practical classes also. It is available by the brand name of Sidex in the concentration of 2% glutaraldehyde. It is more active and less toxic and irritant as compared to your formaldehyde. It is used again to sterilize your surgical instruments like endoscopes, cystoscopes, and other your hospital and laboratory equipment. Then we have phenol, which is carbolic acid. It is the first widely used antiseptic and disinfectant. It was used by Joseph Lister, which you should know he's also the father of antiseptic surgery. And it is produced by distillation of coal tar at a temperature of 170 degrees Celsius. So various disinfectants which come under this are cresol, xylenol, lysol, and orthophenylphenol, which is used in laboratory and hospital settings very commonly as disinfectants. They are toxic and irritant, hence they are only used as disinfectants. Then moving on to antiseptic, again your Savlon, in which your active ingredient is chlorhexidine, and chlorzalinol, active ingredient of your Dettol, and hexachlorophene, formerly used in soaps, toothpaste, but not used commonly nowadays. Then halogen, where we already have studied tincture iodine and iodophore, which is your betadine. Then chlorine releasing agents, which will release chlorine in the form of various ionized forms, like your hypochlorite, chloramines, CaOCl2, and NaOCl form. They are uses to be effective in the free chlorine concentration of 0.5 to 1 ppm. This is parts per meter is calculated according to the soiled, how dirty or how soiled is the particular thing is which you need to clean by the use of chlorine. So chlorine has a particular formula by which you are calculating the ppm which is required to actually disinfect that particular thing. So it is used most commonly in your swimming pools, water supplies, food and dairy products. And 1% NaCl solution is also used as a common household bleach. Then again, your metallic salts, which I've already told you about, they include silver, copper, zinc, lead, and mercury. Most commonly used salt is silver in the form of silver sulfur diazine and silver nitrate. It is the gauze is impregnated with your 0.5% AgNO3, and as already mentioned, it is used as a preventive for burn and wound infections. And zinc compounds are used in your powders and ointments to treat Taenia pedis, athlete's foot, which is actually a fungal infection. And copper sulfate is also an effective fungicide which is used. And thiomycin is a preservative which we are using in vaccine, sera, and other immunological preparations. Then surface active agents, we have cationic compounds which include your quaternary ammonium compounds. They disrupt the microbial membrane and denature the protein. They have a good spectrum of activity against gram-positive bacteria as compared to your gram-negative bacteria, but not your M. tuberculosis or its spores. Example is your alkyl trimethyl ammonium salt and your benzalkonium chloride. And commonly it comes, you would have heard by the brand name of citramide or citavolon. Then your anionic compounds use common soft. They are strong detergent, but they have weak antimicrobial activities. Your amphoteric compounds possess your detergent activity of anionic compounds, and they also have an antimicrobial property of cationic compounds. So basically, they are combining both of your cationic and anionic properties. This particular example is Tego compound. It is used as an antiseptic in your dental practices, but it is known to cause some allergic reactions. Then we can also use dyes like aniline dyes, which includes your crystal violet, gentian violet, brilliant green, and malachite green. These are the common stains which you are using for your gram stain as well as your Zn staining. These are highly selective for gram positive again as compared to gram negative. Similarly, no action against MTB. Similarly. Then we can also have dyes like acrylflavin, euflavin, proflavin. These are also active again against gram positive, but they are not selective like aniline dyes. They have a slightly higher mechanism of and higher spectrum of activity against the other bacteria. Then your gaseous forms includes ethylene oxide. It is used for exposure at 55 degrees for 3 to 4 hours, followed by aeration for 12 hours. Means for the inside this ethylene oxide sterilizer, you will be exposing your product to a temperature of 55 degrees Celsius for 3 to 4 hours. Then you will leave it for normal air to pass through it for 12 hours. And aeration is very important because if you do not do this process, then this gas is carcinogenic. All microorganisms, including spores, are killed by an alkylation process. This is a very big advantage for this because we have very few sets of agents which are sporicidal, the chemical agents which are sporicidal. Next, we have the plasma sterilizer. This is a recent technique as plasma was ascertained to be a fourth state of matter. Apart from your solid, liquid, and gaseous state, it consists of ions, electrons, and your neutral particles. A low temperature plasma by H2O2, paracetic acid, and EM wave is used for this process. Then we have chelating agents, EDTA, heavy metals and derivatives, copper derivatives, a low spectrum of activity these particular set of compounds have. Then factors, this we have already studied. Then how do you evaluate whether your antimicrobial agents are working or not? So the various process of testing of that is agar dilution method, tube dilution method, phenol coefficient or Riedel-Walker test, Schick-Martin test, Kelsey-Sykes test, suspension test, and your in-use tests. So these are your basically tests for carrying out your evaluation of antimicrobial agents. We also studied about the biological methods of indication whether your sterilization process is going okay or not. For that we have hot air oven in which we studied Clostridium tetani. Autoclave we studied Bacillus stereothermophilus. Filtration we use Serratia or Brevandimonas. In your case of ionizing radiation we use Bacillus humulus. In ethylene oxide sterilizers we use Bacillus globigii. In your plasma sterilizer, we use again Bacillus stereothermophilus. Then methods of sterilization and disinfection. These are the commonly available things which are kept in your grand viva tray, and then they are further asked as questions. Ki this particular thing is kept. What is it used for, and how will you sterilize it? So clinical thermometer you would have already seen in various clinics. We are using regular sanitizers based on isopropyl alcohol for disinfection. Then your paraffin glass, syringes, flask, grease, pad, glycerol, we are using hot air oven. For your OT entryway, ward, lab, fumigation purposes, anatomical specimens, we are using formaldehyde, agent of choice. Then for your cystoscopes and bronchoscopes, we are using aldehyde-based agents. Orthoaldehyde is greater action as compared to flutaraldehyde. In your heart-lung machines, your respirators, dental equipments, BiPAP machines, we are using ethylene oxide. For your vaccine, sera, antibiotic, sugar solution, and body fluids, we are using filtration. For your sharp instruments, we are using cresol. Then milk, I already told you we are using pasteurization. For your disposable products like plastic syringes, sutures, swab, catheter, bone and tissue graft, adhesive dressing, we are using ionizing radiations. For culture medias, agars, metal instruments, glasswares, and all suture material except catgut, we are using autoclave. For catgut, we are using ionizing radiation. Then your inoculation wire, which we are using in the labs, we are using sterilizing it by heating it till red hot by Bunsen burner. Infective materials like dressings, bedding, animal carcasses, incineration is used. Metallic surgical instruments, we use autoclave and infrared radiations. Water, we use chlorine in the concentration of 0.2%. Skin, we are using tincture iodine, 70% ethanol, spirit, Savlon. For contact lenses, we are using H2O2. Then this is your common disinfectants and the spectrum of action summarized in a table. So you can see we have divided various disinfectants according to their level of activity against different microorganisms which we have like bacteria, fungus, enveloped virus, MTB, spores, and we have divided it in the various concentrations and according to their level of disinfection, whether it's high, intermediate, or low. So basically, you need to remember your high level of disinfectants are glutaldehyde, 2%, formaldehyde, H2O2, chlorine in higher concentrations. So they are, as you can see, active against bacteria, fungus, unenveloped viruses, MTB, mostly active against spores also. And only chlorine is active is inactivated by organic matter. Rest, all of these are not even affected by the presence of organic matter. So the test which we were discussing about for testing your efficacy of your disinfectants, the first test is phenol coefficient or Riedel-Walker test. It was proposed by Riedel-Walker. It is the expression of bactericidal power of disinfectant as compared to your phenol. So basically, phenol is taken as your denominator, and according to the capacity of phenol, we are evaluating all the other bactericidal agents. This is basically used for specific strains of Salmonella typhi or Staph aureus as a test organisms. So it does not give any indication of disinfecting action under practical conditions. मतलब it is not taking into consideration कि organic matter present है कि नहीं है. It is just comparing your disinfection power as compared to your phenol. So here is this formula. Phenol coefficient is defined as the highest dilution of test disinfectant that is killing your S type in a given time versus your highest dilution of the phenol which is killing the S type in the same time. So the next test we are using is a Chick-Martin test. It is a small modification of Riedel-Walker test. As we studied that in Riedel-Walker test, we are not considering organic matter, but in this case, we are again considering in the act in presence of organic matter. So, but it has a demerits in case that it has it can be used only when disinfectants similar to phenols have been tested. Against other disinfectant, it does not have a good indication. We have Kelsey-Sykes or capacity test. It is used to test the capacity of a disinfectant to retain its activity when repeatedly used with increasing load. So this is your in vitro test which resembles the real life situation and it is used as confirming the it is like the antigen antibody reaction in which we are preparing various dilutions of the test and then we are interpreting that at which titer that particular is acting. Similarly, here we have different dilutions in which we are assessing the particular capacity to which that particular disinfectant will act. So we can use suspensions of bactericidal, sporicidal, and fungicidal activity, and it is both qualitatively and quantitatively measuring that at which particular concentration your particular disinfectant is able to act. You need not know much in details about this. You just need to remember the names. So what we are doing, we are preparing a standard inoculum. That is like I have decided that I will prepare a particular inoculum size of 10 to the power 5 or 10 to the power 6 bacterial concentration. Then we are inoculating it into a specified concentration of the disinfectant. We are incubating it at a particular time and a particular temperature, giving it a specified contact time, and then we are checking for survivors key. After that, with that particular level of disinfectant, what amount of microorganisms are able to survive that particular level of disinfectant. Then the quantitative suspension test is achieved by enumeration. Matlab how many particular bacterial concentration is still there after we have given the optimum contact time and the contact period. So as I already told you, these are not used. These are just you have to learn name basis. We are basically using the in-use test nowadays, which is your Kelsey and Morer's test, which is used on a day-to-day practical basis. It tests the efficacy by determining the ability to inactivate a known number of standard strain of pathogenic Staphylococcus on a given surface within a certified time. It is basically monitoring your performance of the disinfectant when it is used in a real-life laboratory or medical setup. So, a sample of disinfectant solution are taken to determine the survival and multiplication of a contaminating pathogens in a state or over diluted formulation. So what we'll do in this case, we'll transfer 1 ml of the used disinfectant into 9 ml of a nutrient broth. So we are basically preparing a 1:10 dilution into a sterile universal container. Then we are placing 0.02 ml of this mixture on 10 different areas of each on two well-dried nutrient agar plates. So you have first prepared an inoculum in which you have a 1 to 10 dilution of disinfectant and a nutrient broth. And out of this mixture, you are taking 0.02 ml and you are placing it on two different agar plate in which you have 10 different particularly marked areas. Then you will incubate one plate at 37 degrees for 7 days and the other at room temperature for 7 days. Then we'll read if there is growth in more than 5 areas on either plate, then it indicates your failure of the disinfectant. So we are giving it basically two different temperatures and two different settings to evaluate and cover all our bases whether our disinfectant is working properly or not. Then again we have a classification which is a Spalding classification of devices, which is basically classifying your devices according to the level of disinfection